that we do in my lab. So the first thing I have to ask is, how's everybody doing this morning? That's really good, because if you're doing well right now, you're more likely to enjoy what you're about to experience. It's going to bias the way you perceive everything, and that is exactly what we study. All right? How is it that the way you're doing right now changes the way you perceive the world, how you see things as being? I want to get into the exact mechanics of how it works. So let's talk a little bit about what goes on from moment to moment. So let's say you're going around the world and you happen to encounter an object like this beautiful piece of fresh sushi. The wasabi is really fluffy. It looks really good and tasty. Now, before you can do anything about this, your nervous system has to take on a variety of different tasks. Okay, so the first thing you have to do is you have to detect it. You have to have your retina catch all those photons that are bouncing off of that piece of sushi. You have to smell it, maybe even touch it. Okay? Then you take in all of that information from these detectors all over your body, and you start to build together a percept in your mind of what that object is. It's the integration of all these different pieces of information that you've taken in. Then after that, your brain has to make a decision. How am I going to respond towards this object? All right? Then I have to make a plan. I'm going to make this, and then my elbow is going to extend like that, and then my finger is going to reach out like this. And then after that, I have to make all those muscles contract. And I have to actually do that action. So final point here is we eat the piece of sushi. But at every step along the way, our internal state can bias the way our nervous system operates. And that's what makes our brain such an amazing organ, is that from one moment to the next, depending on how we're doing, we can optimize the way our brain operates so that we can do something better if we need to do it better. So let's say that 15 minutes before this beautiful piece of sushi, you ate another piece of sushi at a different restaurant. And that sushi was not quite as fresh. And now that food poisoning is starting to set in a little bit. So our little Vitruvian man changes into this little Vitruvian man. It's kind of, oh, like that. And so maybe your perception changes, that this no longer really can be perceived like this. Maybe it's more perceived like this rotting piece of fish on the beach. And so therefore you change your action. Okay? That's what my lab gets at, is the real mechanics of how this all works. Now, the thing you have to understand about me is that when I was a kid, I was the kid who had very few toys to pass on to the younger kids in the neighborhood, because I just took everything apart. I had to rip off that limb, and I had to undo that hinge. I wasn't a spiteful child. It's not like I was like, yeah, you know, I'm going to break things up. I am a reductionist. I want to understand the little bits and pieces that underlie everything so I can explain it to myself of how this works. All right, so how does our brain do this? How can we achieve this task of changing from one moment to the next? Well, one way in which we do this are chemicals that are called neuromodulators. Now, typically when we think about how neurons communicate to each other, we have neuron A. It gets some sort of input, some stimulus. It has a spike of electricity called an action potential that travels like a wave across the neuron, and it elicits the release of another chemical, which then causes the next neuron to have one of these spikes, a big response. Neuromodulators don't act that way. They simply change the way other neurons will respond if something happens. You can think about it like this. On your stereo, you have a volume knob and you have an EQ adjuster. You can change the left-right balance. Right? There's all these things you can do to change how the music sounds. But you don't get music unless you press the play button. Okay, so that's what makes neuromodulators amazing. You can change the way a neuron operates from one moment to the next, but you'll only know what happened if you have a response occurring. It's what makes our brain such an amazingly flexible organ. Now, the neuromodulator I study is called serotonin. Everybody in the room has heard of serotonin at some point in time. I like this image of a human brain because all these green arrows are showing you where serotonin goes throughout the human brain, and it's everywhere. It all comes from one set of neurons in the hypothalamus called the raphe nuclei. They're all down here. And they project everywhere. Vaguely speaking, serotonin is involved in mood. And you can imagine then, because it goes everywhere, and it's kind of vaguely involved in mood, a large proportion of our treatments for various mood disorders revolve around doing something with serotonin. Maybe we're changing how easily serotonin is made or broken down, or how long it stays out there to signal between different neurons, or maybe it's affecting all the different proteins we have to be able to actually hear the serotonin and respond to the serotonin. So what I want to get at, our lab's goal, is to really understand exactly on a cellular and molecular basis how serotonin will affect the way our brain operates. Now, we do this in these beautiful guys right here, fruit flies. Now, before I go any further, it is not my lifelong dream to cure anxiety in a fruit fly. All right? If you're a fruit fly and you're in my lab, you should be very, very anxious. I have a lot of anxious fruit flies. No, that's not why we study fruit flies, but I will tell you why we study fruit flies. I'm a big proponent of using fruit flies to study the way our brains work. I'll tell you why. The first is that fruit flies are just simply less complex, which makes it a lot easier to answer very directed questions. So our brains have billions of neurons, and each of those neurons make thousands of connections. So that's billions of neurons and trillions of total connections to think about. That is just a very, very complex system. Fruit flies have less than 1 million neurons to work with. Now, you remember I said that there are these proteins that allow us to kind of hear serotonin, to respond to serotonin. You and I have 14 different proteins that allow serotonin to have its effect. And each of those proteins elicits a slightly different response. Flies only have 5 of them. So again, we're making life a lot easier to understand. My goal is to understand the basic principles, and then we can scale those principles up to more complex situations, like in our own brains. The other great thing about fruit flies is they are fast and cheap to work with. I can get through the entire life cycle of a fruit fly in like 4 to 5 weeks, right, which allows us to get this whole experiment done faster and faster and faster. And they take up no space, obviously, because they're tiny, right? And if someone messes up an experiment and we lose a fruit fly, it's okay. No one's too broken up about it. The great thing about this as well is that I have a lot of undergraduates in my lab. So they get the opportunity to go from the very start of an experiment to the very end of an experiment, test very specific hypotheses, and go through the entire scientific method, right, in that very short period of time. Whether or not they pursue a career in science is not important. What I want them to get out of this is an understanding of what goes into the process of the scientific endeavor, and we can do that very easily. And I will note, one of those 4 Mountaineer prospects is in my lab, Savannah Lusk, so we'll see, find out next week. All right? The other great thing about fruit flies is the genetic conservation. So I hate to tell you guys this, but you're all around 60 to 70% fruit fly. It's nothing personal, I'm just telling you a fact. Okay? Roughly 60 to 70% of the genes that allow you to just do everything you do on a day-to-day basis are completely conserved between a fruit fly and us. And what's really amazing about this really speaks to the power of this model system. Almost all of the genes that are involved in the development of the nervous system, fetal development of the nervous system, were found first in fruit flies. Their exact function worked out, exactly what they do, how they operate, who they talk to, all worked out in fruit flies first. And then when everyone had all that information laid out, we go, okay, now let's look in people. And it's exactly the same. So it's so much easier to do it in this faster, cheaper, simpler model, get all that baseline information worked out, and then, boom, it's all applicable to our own lives. That's what's really great about them. The final reason to work with fruit flies are all the crazy cool things you can do to a fruit fly. You can't even imagine the stuff you can do. The sky is the limit. I'm going to tell you only about 2 of these sorts of things, but if you can dream it, someone can do it with a fruit fly. Okay, so the two things I'm going to show you images of from data in my lab. One, we can make select neurons in the brain glow pretty much any color we want. So that allows us to work out the fine architecture of identified neurons so we can manipulate them and see the exact structural changes on a cell-by-cell basis. And we can do it repeatedly in the same neuron every single time, every single day, and it's cheap and easy. The other thing I'm going to show you some data with is that we can make the neurons flash light whenever those electrical signals come along, so that I can now watch the fly thinking, and we can record its thoughts as a visualization. That's amazing. Okay, we can actually just see the thought process, play it back over again, rewatch it. Okay, that's incredible. All right, so back to serotonin. What does serotonin do for the fruit flies? Well, serotonin makes flies smell better. I'm just kidding, they smell terrible. Everyone complains about it all the time, but if I can't make a grammatical joke, then I'm not my father's son. Okay, here is a picture of the olfactory center of a fly's brain. Okay, so think about how small a fruit fly is, and we've now zoomed in all the way to look at just the part of the brain that's involved in the sense of smell. You guys see all those little purple blobs right along here? Each blob is like a unique address for a different odor, or at least different volatile chemicals. So whenever an odor comes along, you get these different blobs kind of being activated in combinations, and that's how you build up the percept of an odor. I'll note, our brain works in the exact same way as this. We just have more blobs. Right? So we have roughly 800 of these blobs. Flies only have 50. It's a lot easier to work with 50 than with 800. I'm the kind of guy that if I have to understand how a vehicle goes forward, I'm going to study a tricycle first, and then I'll move to the Apache helicopter later on. Okay? So what we've done is we've made those neurons that respond to apple cider vinegar. Fruit flies love apple cider vinegar. All right? We've made them glow green, and they'll go right to this one little blob right there. Now we know the address for that odor. Okay? So that's pretty cool. So now let's go ahead and let's see what happens when we throw serotonin in the mix. I'm going to show you an image where I have taken these flies where they will flash light whenever these neurons are activated, and we're going to expose the fly to the smell of the ripened banana. It's called isoamylacetate. So we get a certain pattern of activation, and so when the neuron's really spiking and producing lots of activity, the color is going to look very red. Okay, so here we have this image. Blue means that nothing was excited in this area, but here we go. 1, 2, 3 of these little blobs have started to flash in response to the smell of banana. That's the address in the brain. That's when the fly thinks banana, that's a thought. That's what banana looks like. We put on serotonin. All of a sudden, we get the same pattern, 1, 2, 3, but it's stronger. I've just took the volume button and I turned it up. Okay, so serotonin's making our flies better able to smell. Okay, so where's the serotonin coming from? Now remember I said we have those raphe nuclei in our brain with thousands of neurons that go all over the place. Well, fortunately for us in fruit flies, for the olfactory system, there are just these 2 green cells, which I've made glow green here. I love these cells, they're so beautiful. Here are these clusters with all these little blobs, and they make this beautiful omega shape. Just 2 cells for us to worry about. Now we can start to break it down into smaller bits. We can ask, why are these guys releasing serotonin? And who is listening? What neurons are affected by serotonin? I just showed you that some neurons got stronger and produced a stronger response, right? But why did they do that? Was it because serotonin directly affected them? Or was it affecting other neurons that are then feeding in? Our brains are these complex networks. How do we break it down so we can truly understand how these different chemicals do these different things? That's what we do. So the first thing we start to get at is, why release serotonin in the first place? Well, we know that in our brains and lots of different animals' brains, the neurons that release serotonin are more active when we're more awake. So it's kind of like an arousal state. Okay? So we thought, okay, well, if it's to wake the brain up, how would I control that? If I was going to make a robot, all right, and I wanted to have serotonin be released when the animal is more awake, well, what I probably want to do is get a clock in there, right? So I know what time of day it is. And fortunately, there are clocks in our brains. We have an area of the hypothalamus called the suprachiasmatic nucleus, nice fancy term, but it's basically giving us a kind of an internal timekeeper in our own brain. Flies have the exact same thing in their brains, where they have a set of neurons that keep track of what time of day it is. And all the genes that they need to keep track of a 24-hour cycle like a nice circadian rhythm. We discovered them first in fruit flies, and of course, the exact same genes do the exact same things in our own brains. So here's a picture of the master clock of the fly brain. These areas over here are where the fly takes in all sorts of light information throughout the day. These neurons collect all that information to generate a 24-hour rhythm all throughout the day, so that the fly knows when I should be a little fly who's running around being active, and when I'm going to take my midday siesta, and then when I'm going to be active again, that sort of thing. So we said, let's see if these two sets of neurons talk to each other. So I make my serotonin neurons glow green. I make my clock neurons glow magenta. We look right up close, zoom in, and reconstruct this for you. We find that these neurons are actually very, very closely connected. And so now we've gone further than this. We've found that our serotonin cells are actually expressing the protein so that they can receive the signal from the clock cells. And now we're taking it even further, and we're able to use all the crazy tricks you can do with fruit flies to pull out what genes are being expressed at any given moment by our serotonin cells, to start seeing how these clock cells can regulate gene expression from moment to moment in our serotonin cells. That's the amazing levels that we can get deeper and deeper into trying to understand how this is all controlled. All right, so the next thing I said we were going to talk about is how does serotonin have its effect? All right, so I want to know who's listening to the serotonin from one moment to the next, and I want to understand how they're listening. You remember I said we had all these different proteins. We have 14 different serotonin receptor proteins in our brain, and each of them do a slightly different thing. So I want to know who is expressing which protein and how serotonin is going to affect each of them. All right, so there are many kinds of maps, and that's what I'm trying to do. I'm trying to take an existing map of a brain and add this added layer of complexity. So you can get a map, in this case, of just a satellite view of West Virginia. You can have the counties of West Virginia, or I can have a road map with DC looking terrible like it does every single day. Okay? What I want to build is take that existing map of the brain and lay on top of it an understanding of what serotonin is doing by affecting which specific neuron and how is it having its effect. So this is work that my graduate student Tyler Sizemore has been working on. What I've done here is taken our West Virginia colors. In blue, I've colored in those neurons that express a given serotonin receptor. That's what we do. We can make them glow, so we can identify those guys. And then we can go, okay, I have different tags that let me to identify each and every single cell type within this circuit of the brain, so I can try and identify who is expressing what receptor. What's cool is that this receptor, I've shown blue here, and I'll get this going as well. Oops. It is an inhibitory receptor. If serotonin binds this, it will suppress the activity of these neurons. Okay? Yellow are the neurons that are themselves suppressive. All right? So what I'm doing is something called disinhibition. It's like if you have to choose how to make your car go forward, you can either put your foot on the gas or take your foot off the brakes. What serotonin is essentially doing then is taking your foot off the brakes and letting things roll forward from there. So that's just a basic operating principle that we can pull out from this to see if that's applied to more complex circuits. So taking a step back from this, your brain is an incredibly flexible organ. Right? You can do the most amazing things with your brain from 1 second to the next. It's not like you have a second brain for hunger and then a brain for starvation. I have a simple experiment I'd like you to all try over the next 2 months. Okay, we can do this together. Every single Tuesday at 6 p.m., that's when I want you to go grocery shopping. Okay? I know this is science, so it's really important. Okay? We're going to make this work. Okay? So at 6 p.m., you're all going to go grocery shopping. However, on odd number weeks, I want you to eat your breakfast, your dinner at 5. On even numbered weeks, I want you to eat your dinner at 7 after you've gone grocery shopping. So half the time you're going grocery shopping when you're hungry, and half the time you're going grocery shopping when you're full. I want you to take a look at your bill between those two situations. Now, your grocery store has not changed. You don't have a hungry grocery store and a full grocery store. The prices haven't changed. The products probably don't change that much from week to week. But your own perception of the value of those food items changes based on how you are currently doing. And we're talking about making decisions about getting enough food for the whole week. So this isn't just immediately what am I going to eat right now. It's for the whole week. Okay? It's this fine-tuning that allows us to do that. And so our goal, all the students in my lab who get all this great data for me, is to understand the mechanics of how our brain pulls this off. And so with that, I'll open up for any questions you may have.
Do you get grants to do the research?
Oh yeah, I'm funded by the National Institute of Health to exactly figure out this whole map of understanding which sets of neurons within our network express which serotonin receptors, so we can start delving apart into the exact mechanics of how it does what it does. So our next goal, now that we have this map, is we can selectively go into those identified neurons and remove that receptor and ask, now what happens to the network?
So you've been doing this a number of years and you're
Oh,
a
yeah.
young guy. So where do you see all of that going?
Yeah, so the end goal for me is to have a set of operational principles that we can then see that, okay, well, we see that serotonin is operating by doing these 3 or 4 strategies. And is this true in the spinal cord? And is this true in auditory cortex? And are these principles that are applied over and over and over again? And then we can ask ourselves when things go awry, hey, you know, that kind of reminds me that this is a deficit in this system. This looks like that principle. So now we're going to have a more targeted approach instead of just going ahead and hitting all receptors. The thing that's so cool about this system is that I can go out and pull out that receptor from one type of neuron. But there are thousands and thousands and thousands of neurons that will express that one receptor type. If I were to just get rid of that receptor or just activate that receptor, I would be having a huge slew of different effects. Not necessarily all of them the ones that I really want to be having. So by understanding all the basic mechanics of how this is all laid out, then we can take a more targeted approach to things rather than more trial and error. Yeah. Yes?
Andrew, I had the opportunity to visit your lab and actually look into the microscope, look at the brain of a fruit fly. Tell me what your undergraduate students, what you challenge them to do each day with respect to.
So it depends on the students. We always you have to start them out kind of really the ground floor, and then you build them up on their strengths. So some students can go in there and they have no problem dissecting out a fruit fly brain. After a week, they can do it with these tiny little forceps, and they can actually get the brain out and it looks beautiful, it looks totally intact. Other students, it takes a very, very long time for them to get that. So what I generally like to do is start them out with a graduate student mentor who can really guide them, and they'll start out with very straightforward sorts of things to do. Maybe it will be analyzing pre-scanned tissue. So we can do cell counts, things like that, and they can start to get a directed approach from there. And once they've mastered that, we add more and more and more detail to it. So I can kind of build them up and play to their strengths. Because each student is different. You know, there's some students who come in with, we call them lab hands. And these kids are just, it's just amazing what they can do. They can just do the most fine motor skill stuff you could imagine. But others just simply don't have that, but they're very inquisitive and they want to be able to have these sorts of experiences. So it's really a student-by-student basis. You start them out with a simple little project. You know, how many of these cells have that sort of thing? What happens if we take these flies and we get rid of that protein? What happens to their behavior? Can we put them in these little, we make tons of little fun arenas for the flies to do stuff in, right? So you go ahead and they go through and they say, okay, counting up the fly's behavior has been messed up in this way when I take out that protein from that one set of neuron. Right? And so they build up and move from there.
So you challenge them in terms of expanding that experimentation process?
Oh yeah, yeah. I mean, they are contributing towards primary literature publications every time. I just had a paper come out in the Journal of Comparative Neurology. It'll be getting the cover for the April issue. And the third author on that is an undergraduate student in our lab. So he will have, be an author on a publication. And most of my students are collecting real data towards publications. This is not an exercise. This is all collecting primary literature. They also all write a proposal, right? And then they have their full thesis that they have to defend. Sometimes I'll take students with me to conferences, and they'll be presenting their work as posters at an international conference to very, very high-ranking professors from a variety of different universities all over the planet. So they get the full experience. They also get the full experience of when the experiment completely fails, and you're just left with the ashes of all this hard work trying to figure out what in the world went wrong. And they get to stand back and go like, oh my God, what, could it have been this? Could it have been that? And they have to figure it out. And that's part of science. So it's good for them to get that opportunity and to see that science does involve a certain amount of luck and just making it all work. Yeah.
In your studies reveal why some people are more predisposed to get brain cancers?
Oh no, this is all, for me, this is all very basic mechanics of how it works. So for me, the most important thing to consider is that biomedical research occurs on many, many, many different levels. We're talking all the way down to just understanding how things work in the first place in a healthy, normal brain. I shouldn't say normal brain, but at least a healthy brain. All right, and then building up to more and more complex systems. So for me, it's not about figuring out how a disorder works. I want to understand the mechanics of how things normally operate, and then from there, let other people apply those principles to more complex systems. Yeah. Yes?
Are most of your students destined for med school?
I would say probably 75% of my students are going to med school, yeah. But there are a good proportion that go to graduate school or take on technical jobs. For me, it doesn't really matter where they want to go. If they're excited to be part of the team and they want to see what it takes to get through a scientific study, that's all I care about. That's what I want them to have that experience. Because we see studies in popular media, oh, the popcorn odor, the smell that they add as chemical, it's cancerous, it's the worst thing ever. I'm like, well, really, what's the data behind that? How do they go ahead and build that whole study? Because you want people to be able to make informed decisions and not simply get a little soundbite that they just digest and blindly follow. I want them to think about how that data was collected. And I want them to have that experience. Yes?
Andrew, what are the potential medical/behavioral applications of this research in humans?
I mean, for me, I think having a complete understanding of how the nervous system operates is extremely important. Up until a few years ago, there was always this dogma that, yeah, if neuron A releases chemical A, it will always release chemical A. Turns out that under certain conditions, we can make neurons switch which chemical they communicate with. That is, as an experimentalist, that's horrifying, right? Because you try every single day, you're like, okay, we got to control this, we got to control that, and now the neurons can switch which chemical they're releasing? Come on, give me a break. Something's got to go my way. How could you have this happen? But the idea is that we can get all this continued information of how we go about doing these sorts of things. And then with that knowledge, we're better armed and prepared to try and develop therapeutics later on. But if you don't understand where you're starting with, you don't know where you're going. Does anybody here listen to Car Talk? I love Car Talk. That show is amazing. You can call those guys up and you can imitate what your car sounds like, right? And they'll tell you what's wrong with your car. Those guys understand how a car works, right? So we have to, the brain is obviously way, way more complicated than a car, all right? So we need to understand everything we can about how the brain works, to be able to make these educated decisions about how to fix problems. And so, that's the strength of using an animal like a fruit fly, is we can get these really basic questions of how this all works, and then scale it up from there. Yeah. Yes?
I don't really know the life cycle of a fruit fly, so is this really talking about more an adult brain works, or do you have it developmentally?
Yeah, we stick with, I'm generally sticking with the adult flies. That's a whole field of other people who are doing similar sorts of things, and so we're always kind of comparing notes and thinking about all these sorts of issues at all these different stages. But yeah, lots of people are studying development of fruit flies as a way to look at how our own nervous system develops. I mean, all the genes that are involved in deciding that your neck goes here, and then your shoulders are here, and then everything goes in a certain order. All those genes were first found in fruit flies, and all those genes are laid out in the same order in a fruit fly as they are in your body. It's amazing. And what makes you have a front in front and a back in back, all discovered first in a fruit fly. And then it's all just found to be true in us. All the genes that allow us to have that 24-hour rhythm, right, that's completely entrainable, given all sorts of clever names. Fruit fly people, we love giving clever names to things. So these genes are called like clock and cycle and period and timeless, right? Haha, because it's about the day. But all those genes, they do the same thing in our brain as they do in a fly's brain. So it's just a higher throughput system where you can get the experiments going just so much faster, figure out how it works very cheaply, very quickly, and then you go ahead and you can apply it. But if you were to start at a mouse where it costs $5 a week just for one mouse, right, as opposed to me having tens of thousands of fruit flies for like a penny a week, right? You just scale things up a lot faster. But you have to have model systems and levels of thought all throughout the scheme of things, from the most basic level to the most applied. You can't jump straight and just think about applied. You're out of luck if you do it that way, because then you're just guessing. You have to have everything built upon everything else. Yeah. Yes?
All fruit flies have the same amount of serotonin? If they don't, why don't they? The impacts of that?
Yeah. So you can imagine that there could be certain conditions where you could see that there might be variation. I mean, for all intents and purposes, I really don't know the answer to that. It's hard to be able to measure the amount of serotonin in a single fruit fly's brain to see if there's variation between one fly and the next. But you could imagine you could have some sort of, that could be something that you could assay, right? Let's monkey with this or set up these sorts of conditions and see if that affects it. So one thing we do in my lab is we use a very clever approach to remove the enzyme that produces serotonin in our cells of interest, so they can't make it anymore. And so what we find is, what we're trying to figure out is, does this cause the same thing as if I shut down the neuron, as opposed to make it still just as active as it ever was before, but now it can't make serotonin. And so these are the sorts of approaches you could take to try and see what the role is. So I'm glad you brought that up, because that's the exact experiment we're starting this week, is to try and hit just our two cells, leave all the other serotonin in the entire nervous system intact, but in just our two cells, we take out that enzyme, can't produce serotonin anymore, and what's the impact? So what we found, where we take those two neurons and we make them unable to produce those spikes anymore, is the flies, it's kind of like they have a stuffed nose, right? They have a very, very hard time finding food odors at low concentrations, and this is work that my undergraduate student, Sarah Michaels, did, and she won the best undergraduate poster at the WVU Summer Undergraduate Research Experience last summer for this work. But she found that if she made those neurons unable to produce an action potential, flies just couldn't find the odor that the apple cider vinegar that they normally love. If you have it like pure high concentration, they can find it, because it's like asking someone if they can, if you've got glasses on or not, you can find a firework if someone blows it up in front of your face, right? But at that lower concentrations where the flies have to work a little bit, they essentially become anosmic. So they lose that sensitivity. And so yeah, now we want to go ahead and do that and just remove the ability to produce serotonin to show the same sort of effect. Yes?
Well, that leads into my question. It sounds crazy, but do you think it's a digital system or analog?
Oh no, I think it's analog. I think it's analog. So the question is this digital or analog? I don't think serotonin is just turning something on or turning something off. It's all volume control. And the other crazy thing with all of this is, I'm talking about just serotonin, but it isn't just serotonin at one given moment. There's also dopamine. And there's also going to be some release of acetylcholine. And then you have circulating hormones like cortisol and then insulin. Everything's happening at the same time. So it's all one big soup. And that's what's dictating what each of you are experiencing from one moment to the next, right? Because you may have all just eaten all had breakfast roughly the same time today, right? And you had roughly the same sorts of things. But maybe you all went to sleep at different times last night. Or maybe some of you had just a knockdown fight with your spouse last night. And that still is just eating you up, So you are each experiencing a different kind of chemical existence right now, and that all plays into how your brain operates. I'm being told we're out of time, though. Is that true? All right, well, thank you very much. I really appreciate all of your questions and your time. Thank you. Yes, if we can find our seats and